In this video, we're going to talk about the forces that drive protein folding. And we're going to see that the primary structure or the sequence of a protein will dictate its three-dimensional structure. We have to remember that proteins don't fold in a vacuum. They fold in an aqueous environment, and that aqueous environment is critical to them achieving their native conformation. So here I'm showing you a molecule of RNase A. And RNase A is a small protein that is a classic model system that's used by protein chemists to study the process of protein folding. Now I've added thousands of water molecules, so the protein is now in its aqueous environment. It's surrounded by water molecules and also a lot of other ions like sodium and chloride, for example, that are illustrated here. And these water molecules interact with the protein. They do so transiently because they're tumbling in solution, but these thousands of transient interactions are critical to the process of protein folding. Let's consider the forces that dictate how proteins fold. And so now I've gone to a schematic diagram that illustrates an unfolded protein with all the little colored spheres as the side chains of the amino acids. And then when the protein is unfolded like this, it will have many, many different conformations that it can take, and these conformations can interchange. And that means all that conformational flexibility provides the unfolded protein a lot of conformational entropy. Now, once it folds, it will achieve one single native conformation, and that greatly reduces the conformational entropy of the protein. And it turns out that this is the major force against protein folding, so that it opposes protein folding because you have a loss in entropy. So this is where the water comes into play. Free water molecules will tumble in solution, and in all the different orientations, they can see other molecules and form hydrogen bonds. So they have a lot of conformational entropy. Now we can contrast this to a water molecule that's on a hydrophobic surface. For example, the hydrophobic surface of a hydrophobic residue, a side chain that's exposed to solvent. Now that water molecule will have fewer conformations or rotations that will enable it to have the same number of hydrogen bonds. And so it will have reduced conformational entropy in comparison to free water molecules. So now if we return to our schematic of protein folding, we can see that upon folding, a lot of the hydrophobic side chains, which are the little yellow spheres in our diagram, are buried in the hydrophobic core of proteins upon protein folding. And that means that now a lot of the water molecules that were interacting with those side chains are now free to interact with other water molecules. So there's a great increase in the conformational entropy of water upon protein folding, and actually of the whole system. So there's a favorable gain in entropy in the system by releasing water molecules from hydrophobic surface when you bury them in the hydrophobic core. And this is called the hydrophobic effect.
So are hydrophobic residues always buried in the hydrophobic core of a protein?
Actually, not all of them, because about half of the residues on the surface of proteins are also hydrophobic residues. And these residues are often important for interactions with other proteins, with ligands, or with substrates. And another situation where we find a lot of hydrophobic residues also on the surface of the protein is in membrane proteins, where these surface residues will interact with the hydrophobic lipids in the membranes. So let's get back to the hydrophobic effect. Another reason that the hydrophobic effect is really important to protein folding is that it's a highly cooperative process. So if you imagine two hydrophobic side chains that will start contacting each other, they constrain the backbone that surrounds them and will lead to other hydrophobic side chains now being in close proximity and also interacting with each other. So you get sort of a snowball effect, and the hydrophobic core will zip up rapidly to hide all these hydrophobic side chains on the inside of the protein. However, recall that the peptide backbone contains polar amino and carbonyl groups. How are these groups buried inside the hydrophobic core of proteins? As we learned in earlier videos, hydrogen bonding within the main chain stabilizes secondary structures like the alpha helices and beta sheets. And these hydrogen bonding patterns also serve to balance those partial charges on the polar groups of the peptide bonds. And that is what allows the polar groups to be buried within the hydrophobic core, because they're already hydrogen bonded. And similarly to the hydrophobic effect, the zippering up of the secondary structure elements is also highly cooperative. These non-covalent interactions that form within a protein, including also the salt bridges and the van der Waals interactions, are the final major contributor to the energetics of protein folding. They contribute to a favorable and net decrease in the enthalpy of the protein as it folds. So now that we know about the three major forces that drive protein folding, let's put them together. So I'm going to show you some estimates of the energy contributions from each of these forces to folding a protein of about 100 amino acids. It turns out that the average size of a domain, the independently folding unit of proteins, is about 100 amino acids. So the hydrophobic effect due to the burial of the hydrophobic amino acids into the core of proteins will yield minus 140 kilojoules per mole of protein upon folding. So that's a favorable energetic component. The second favorable energetic component are the non-covalent interactions, which provide, again, about minus 150 kilojoules per mole of stabilization energy. Then the forces that opposes these two is the huge reduction in entropy from going from many, many conformations accessible to the unfolded protein to the single conformation of the native protein. So when we add up those three components, we still come out with a net favorable free energy with about minus 70 kilojoules per mole of stabilization energy upon folding of a 100 amino acid protein. And that means that the process should be spontaneous. These numbers are estimates, and they are done with a few simplifying assumptions, but it turns out that they agree quite well with experimental results. So it turns out to be a simple matter of thermodynamics. Protein folding is thermodynamically favorable. Still, you can see that the net effect is rather small because we have a few large forces that almost cancel out. So now we know that thermodynamics favor the folding of proteins in an aqueous solution. But I mentioned earlier that there's one native conformation to proteins. How do the proteins reach that one single native conformation?